Good afternoon, everyone. My name is Joe Devaney. I'm the director of the Laboratory Innovation Team at GeneDx. I'm very excited to be here and to talk to you all. I'd like to thank Illumina for the invitation and GeneDx for allowing me to come. I'm going to talk about mapping the future of whole genome sequencing with Illumina constellation technology. I do have some disclosures. So just an outline of what I'm going to talk about. I'm going to discuss a little background. Then I'm going to talk about a research study that we did comparing results we had from our molecular lab to long-range sequencing with Illumina's constellation technology, different sample types, and then different variant types. And then I'm going to give a little bit of a summary of the work. So I just wanted to take a step back and talk a little bit about the diagnostic odyssey. It's common, it's critical, and it's costly. And you know, these are some very sad statistics to talk about, but oftentimes a pediatric patient will wait up to 5 years for a diagnosis. They might undergo up to 5 uninformative tests, and of course that's going to cost a lot of money. At GeneDx, we really want to eliminate this. We want to shorten the time, and we want to shorten the amount of tests that we have to do to get an answer to that question for that patient. There are some key challenges and possible solutions in rare genetic disease. One of them is that there's causative variants maybe in genes that we just don't know what they do, or we don't know what the function is. So we might be finding those causative variants, but we can't actually assign that it leads to any kind of disease. Causative variants can be very difficult to detect. As was mentioned in the beginning was structural variants. How do we detect those? And that can be difficult to detect with short-read sequencing. There's also some new exciting sequencing technologies, long-read. However, these have to be, we really have to dig in and make sure that these work in our lab. And then one other thing that possibly stands in the way of using that is that it requires new instrumentation. Whereas long-range sequencing or constellation, we can use technology that we already have in our lab. A lot of labs have NovaSeq X Pluses or NovaSeq X's, and this technology can be used on that. So I just want to talk a little bit about the samples that we selected for this study. I consulted with our lab director, Kathleen Haruska. She's fantastic, and she and also some of the clinical directors, I went to them and I said, I really want to look at the difficult variants. What takes the most time? What is what's like the toughest variants to see? Then we also wanted to look at variants that were in those problem exons. That's from the Wegener paper in 2019. We also wanted to try and dig into samples that were inconclusive with the current methods that we do. And then also I wanted to have a mix of sample types. At GeneDx, about 60% of the samples that we get in-house are buccal swabs. So I wanted to make sure that we could look at numerous different sample types. And I also have a little table of the original diagnostic method and also the combination of diagnostic methods that were used to look at these variants. Just a little bit about the samples that we used in the study. There was about 58% female samples. One sample was unknown. It was a prenatal sample. We also tried to look at, like I said, numerous different sample types. We used blood. We used high molecular weight extracted blood, buccals, prenatals, and then we also used samples that were extracted in a lab outside of GeneDx and then sent to GeneDx for analysis. So one thing that with using these different sample types that we examined, we really saw that DNA quality directly correlates to the template length. And I'm going to show a chart of all the different sample types and what that data look like. But needless to say, the buccal samples don't perform as well as blood. Prenatals, some of them performed very well. We have a couple different extraction methods we have to look at. And then also the external DNA, which we don't have an idea how it was extracted, but that performed okay. So to kind of dig in more to that sample type question and what the answers we can get from the constellation data, really we want to look at 3 different pieces of data. One is the percent above 60 kb. The other is, sorry, I'm having a hard time seeing it, but it's the template genomic length, and then also the fully phase genes. So as you can see at the top, for the blood extractions, the glass bead and the magnetic disk extractions performed well with the greater than 60 kb. Also, the template length was higher, and then we got fully phased gene information on up to 92% of DNA samples from blood using the glass bead method. We also did, as I mentioned, buccal swabs. We've used a couple different extraction methods for these buccal swabs, so I don't really have most of those methods were all magnetic beads. Again, you can see the percent 60 kb, and then the template length, and then how much of the genome was fully phased. The prenatal samples, the only method that we see is a potential problem is the salting out precipitation. I would comment that most of these samples were all cell lines that were grown from a prenatal sample. So we hypothesized there might be something in the buffer that we might need to do buffer exchange and clean that up. But the relationship between the percent greater than 60 kb, the template genomic length, the higher those are, the more percent coverage you're going to get in a genome. And this is a list of all the different variant types that we looked at. We really tried to cover a whole plethora of different variant types that we could. I have some little asterisks or like plus marks and to show. And just to comment on those, PKD1 is a gene that we're still working on. So we looked at a deletion in PKD1. We also looked at phase of a PKD1 deletion and a variant. So we're still working on that. It's a work in progress. Mitochondrial DNA deletion, we're still working on that also. I should say Illumina is working on that. But we were able to detect heteroplasmic SNVs in mitochondrial, and the percent heteroplasmy matched what we had seen in our testing in our laboratory. The other sample type that was kind of interesting to look at was we had some triploidy samples, and we could visibly see they were triploidy. But they don't have a calling method yet for that in Dragon. And we had FSHD contraction samples, and they don't have a method yet supported in Dragon for that. But I'm sure they're working on it. So a little bit about the workflow. Again, this was already explained earlier. I won't go through this. And I'm sorry to say these are pictures of me in the lab, which I will say that when I came in, I work in a lab, but when I came into the lab, my fellow lab mates were all like, "Why are you here? Are you going to ask us a question?" And I said, "No, I'm going to do some work." So I actually did process some samples for this method, and the reason that I could do it, it's very, very easy. It only takes about 10 minutes. There's 3 reagents that are added together, again, it's a very simple process. We use 350 nanograms of DNA, although we're going to test going down to lower levels of DNA. Sequencing, it's a standard NovaSeq X Plus. It's a custom recipe, 2x150 sequencing run that takes about 24 hours. And then we use Dragon for analysis. We get the standard outputs from Dragon that you get for a short-read genome, but there is some new output files which I'll show a little bit of. Some benefits. As I mentioned, this is a really easy process to do it. No thermocyclers, no robotics, which makes me happy. I love our robotics team, but whenever we ask them to do something, there's always, it's not an argument, but it takes some time. And as I mentioned, it's compatible with multiple sample types. There are some challenges, though. One is that it's lower throughput than what we get from standard sequencing. There's also a little bit less flexibility. So on a 10B, we can run 8 samples. There is, again, as I mentioned, some of the new outputs that we're going to have to learn how to look at that data. So I'm going to jump right into the data that we saw. First of all, we wanted to look at some low-mappability genes. For example, spinal muscle atrophy, spinal muscular atrophy. SMN1 is causative. SMN2 is a pseudogene, shares about 99% homology. For Lynch syndrome, PMS2, is the gene that causes Lynch syndrome. The pseudogene share up to about 99% homology. Chronic granulomatosis disease, NCF1. NCF1 has 2 pseudogenes that share over 99% homology. And then recessive non-syndromic hearing loss is another phenotype we wanted to look at. STRC and its pseudogene share, again, 99% of homology. So a tool that was developed specifically for this constellation work is called multi-region joint detection, MRJD. I'm not going to try to walk through what it is. I mean, I'll give you like a rough idea, but trust me, I'm sure I'm going to get stuff wrong. Basically, the proximity information from all the reads are joined together. And so the way I want to think about this is, and I know this is probably going to get me kicked out of the city, but region 1 I'm going to call the Yankee gene, and region 2 I'm going to call the Red Sox gene. It's obviously the pseudogene. I told you, I'm going to get kicked out. I'm sorry. I better get a lift real quick. Anyway, so the information is pulled together. Then you can do copy numbers based on all these reads that are mapping not only to the, I'll say region 1, region 2. You can get copy number. And as you can see, the Yankee gene had more copies, which is great. But then you can use haplotypes and variants within those copies to discern the haplotypes. So this is a really useful tool, and the output is phased genotype data. So with this, SMN1, we're able to detect a sample that had 0 copies of SMN1. We're also able to detect the number of copies of SMN2, so all based on the variant that exists between SMN1 and SMN2. This is useful because not only do you see that this individual has spinal muscular atrophy, but then you can also discern how many copies of SMN2, and that can be used for therapeutic treatment. PMS2, this is a variant that we looked at where, and I'm not saying anything about our lab. We call this a deletion in PMS2. When we actually dug into the data, you can see the nice haplotypes for PMS1 or PMS2, haplotype 1, haplotype 2. You can see it's actually a conversion between the pseudogene and the gene of interest, and there's no copies of the pseudogene. So this is something that we're going to go back and talk to our clinical director and see what we can do about this sample. One of the other genes I mentioned was NCF1 and its 2 pseudogenes. I'm just showing what the short read coverage is for those 3 genes. There's a variant in exon 2. It's a GT. It's in the gene. The pseudogenes both have a deletion of that GT. And I highlighted where exon 2 is on the gene and the pseudogenes. You can see that the pseudogenes have almost no coverage in the short read genome. But using MRJD, we're able to discern the haplotypes of NCF1 and the pseudogenes. And with this information, we were able to look at a homozygous variant in NCF1 and map it correctly to the NCF1 gene. Able to look at loss of 1 functional copy, 2 functional copies, so complete deletion of functional copies of the gene. And then we had this interesting sample where we saw 6 copies of the gene, kind of like the image I showed where it showed the NCF1 and the pseudogenes. And it turns out that this is actually a conversion. All 6 copies had the delta GT. All were missing that GT genotype. STRC, this is a sample that we ran on a panel. And then we also used MLPA to detect the deletion. We saw the deletion. We don't actually do sequencing analysis on panels for STRC, but using constellation technology, we were able to find a nonsense variant. So we saw 2 hits for this particular sample. Repeat expansions, I won't go into this, but I will say that we compared long read methodologies, what the sizes we got, to what we saw in constellation. Pretty comparable. CNVP, which gets extremely large, we saw relatively good concordance between the size that is pathogenic. And that's really the important thing we're looking for is the Complex structural variation, these are co-location plots. I love looking at these because you can see a nice image that tells you kind of what it is, a deletion, an insertion, inversion. And we looked at a pericentric inversion. We were able, this is soft clip reads, and using the co-location map along with high glass visualization tool, you can actually pick out that inversion, and you can pick the exact region and where it's at. This is a small inversion in IDS. It's a 30 kb inversion that we were able to pick up. Again, one interesting thing about this is we ran this on a short read genome and we did not see a call in structural variants, or we couldn't really see it in the BAM files. So this is a case where Constellation gave us an answer that might not have been picked up with the short read genome. I'm not going to go into a lot of detail on this, but this is an extremely interesting sample that we had that had multiple deletions and duplications in chromosome 18. Using Constellation, we were able to see the ring structure, and there's a multitude of data that went into this, but it was fascinating to see it. So a little summary of what we saw with the Constellation technology. We saw 99.5% variant detection. We did have some misses, PKD1, like I mentioned, mitochondrial deletions, and then there were a couple of variants that they're not callers for now, but like I said, hopefully they're going to start working on those. We saw reproducible and accurate results with different sample types, including blood, buccals, prenatals, and next we're going to try some dry blood spots. We really want to see what this technology can do with all the different sample types we see at GeneDx. We did call a mosaic trisomy that we missed with a long read method, and we also saw the ability to call DNA heteroplasmy, again, the percent heteroplasmy for single nucleotide variants and indels. It matched very well with our current lab technology. And again, the nice thing is that this technology combines numerous lab technologies into one, and hopefully that'll shorten the TAT. And so just some overarching themes about this. It is a really simple workflow. I did it. If I can do it, I'd say that 99% of people in the lab can do it. We did see a significant enhancement in the detection of structural variants. In fact, there are some differences in what we reported and what we saw, so we have to discuss that. Our lab will discuss that. And the other nice thing, I didn't really talk about this, but we saw nice phasing information. We saw phasing information for SNVs, indels, repeat expansions, and also CNVs, and up to about a megabase between the two different variants. So really, I think Constellation is a pretty powerful tool. I'm excited to keep exploring it and see what we can do with it. I do have some comments about what's needed, and after Beckham's talk, I mean, right, methylation data has to be there. And also the scalability, right now we run 100 genomes on a NovaSeq X Plus, and we'd have to drop that down to only 16. So there has to be some work done on that. And as I mentioned, there's some difficult gene regions, mitochondrial DNA, other things that we need to look at. So, but I'm sure they will be worked on.